phase, living its cell life in G1, and then it gets a signal. It's time to divide. This is a non-sex cell, somatic cell. We're doing mitosis. So it gets a signal, it's time to divide. The first thing it does is it goes into the S phase, replicates DNA. So it replicates each of the homologous chromosomes, right? So what has to happen in replication? Well, we said we have double-stranded DNA, and we said what enzyme opens the DNA? Helicase. What enzyme is going downstream and keeping it from getting twisted up? Because if think about like when you untangle hair, there's two words for it, two names. Gyrase is one, and I think about how it gyrates, so it keeps it from getting twisted. I don't know if that helps, but topoisomerase is the other one. I usually use gyrase because it's easier. Same thing. Keeps it from getting twisted. So helicase opens up the DNA, right? DNA polymerase doesn't know where to go. It needs some primers. So what lays down the primers?
Okay.
So what would be a good enzyme name for something that puts down primers? Primase. So what do you think it's called? It's RNA primase, though, because it lays down RNA primers. And one of the reasons it's important that it lays down RNA primers is because there's an enzyme later that comes back and takes those out, and it can recognize the RNA easier. So I've opened it up with helicase, keeping it from getting twisted with gyrase. I'm looking to keep it open because DNA wants to be double-stranded. What do we use to help keep it open and stabilize it open? So what would be a good name for something that wants to keep single-stranded DNA from binding together? Single-stranded binding proteins. They're just like doorstops. They keep it open, right? So they keep the DNA open long enough that the enzymes can come do their job. So we have it open. RNA primase lays down an RNA primer. Now DNA polymerase goes, oh, okay, this is what I do. So DNA polymerase comes and binds onto that primer. And then DNA polymerase can only go in one direction. What end of the growing strand of DNA does DNA polymerase add nucleotides to? 3 prime. It can only go one way. So then you have a problem. One of the strands is building the complementary strand 5 prime to 3 prime. That's easy. Keep adding nucleotides, go forever. Find that primer, go forever. The other strand, the complementary strand, is running from 3 prime to 5 prime. So it's running 3 prime to 5 prime. Well, DNA polymerase can't add that direction. Like it's growing that way, it should be. So what it has to do is it has to go to a primer, go in the 3 prime direction, jump back to the next primer, go in the 3 prime direction, jump back, go in the 3 prime direction. So it's copying it discontinuously. And we call that the lagging strand. The other strand's the leading strand. Binds the primer, goes all the way straight through. So in the lagging strand, we have an issue because now we have multiple primers and they're RNA. So there's an enzyme called DNA polymerase 1 that goes in and pulls out the RNA primer and replaces it with DNA. And then we have pieces. And we have an enzyme that ligates or glues the pieces back together. What do we call that enzyme? Ligase. Puts them all together. What are those fragments called? It's a fun word. Dr. Okazaki. Right, so Dr. Okazaki, they're little Okazaki fragments. Okazaki, think about I think it's just fun, right? So it glues those pieces together so that now you have 2 double-stranded pieces of DNA when you get done. So you've replicated that. And then to make sure we're clear, when we started, we had 2 copies of the same chromosome. What do we call these? Homologous chromosomes. Why are they homologous? They have the same what? Genes. So if there's a gene for awesome on homologous chromosome 1, there's a gene for awesome on homologous chromosome 2. They can be different forms, little a, big A, whatever, but they have the same genes. So I have 2 homologous chromosomes. What do we call that when a cell has 2 copies of the chromosomes? Haploid or diploid? Diploid. Diploid means 2. So diploid, somatic liver cell says, I need to divide. First thing is it does, it goes through all that replication, and it replicates these homologous chromosomes. When it replicates them, what do we call this? Sister chromatids. Are the sister chromatids genetically identical? Yes. They are. They should be, unless there's a weird mutation. DNA polymerase is pretty good. It's high fidelity. It doesn't make many mistakes. Are the original homologous chromosomes genetically identical, base pair for base pair? The original ones? The one from mom and the one from dad?
No.
Hopefully not, right? Unless mom are genetically identical twins, then the original homologous chromosomes, even though they have the same genes, they're not genetically identical forms of the same genes. One.
In meiosis, when that happens, there will be 2 different colors on each side, right?
That's after crossing over.
Okay.
So we have replicated our DNA. So now I have homologous chromosome 1 with sister chromatids, homologous chromosome 2 with sister chromatids. So during prophase, they pile up. During metaphase, they line up, right? So here is my little metaphase. Here's my 2 cells, right? So I've got my metaphase plate. And what's going to separate during metaphase? What am I going to line up and separate during mitosis? Am I going to separate the homologous chromosomes, or am I going to separate the sister chromatids? Sister chromatids. Because if you think about it, I want to end up with 2 genetically identical diploid cells. I want my liver cell that I made to look, because basically I made 2 now, right? I split 1. Those should look genetically identical, and they should look just like the parent cell. So that is, now I have 1 pair of homologous chromosomes over here, 1 pair of homologous chromosomes over here, and I have 2 happy cells living their happy cell life. For meiosis, I have some kind of a stem cell, right? It could be a sperm stem cell, it could be an egg while you're developing in utero, whatever. That original stem cell is diploid. It has 2 copies of the homologous chromosome. What's the first thing it has to do before it goes through meiosis? Same thing. It has to replicate the DNA. It does the same thing it just did. I now replicate the DNA.
Is it bad if I got the
No, just wash your hands with water. You're good.
Okay.
Soap and water. So now I have sister chromatids that are genetically identical, sister chromatids that are genetically identical. But something cool happens in meiosis that doesn't happen in mitosis. So these all come together and form a tetrad. Tetrad means 4. Call it like a synaptosome. So they're coming together. One of the things that happens is that in meiosis, crossing over occurs. So this is where the non-sister chromatids can switch some genetic material, right? That's called crossing over.
Who was it again?
So now, these are all 4 together. They're going to line up on the metaphase plate, right? Now I have, you notice, they're not genetically identical anymore, right? I've done crossing over. And the non-sister chromatids cross over because what would happen if I cross over red and red? They're identical. They wouldn't do anything, right? Biologically, there's no benefit to that, right? And there's really cool stuff that happens when that crosses over, but it's more than we have to know now. Okay, so I'm lining up on the metaphase plate. And in meiosis, the first meiotic division for meiosis 1 is what we call reductive division. We go from diploid to haploid. Diploid means we had 2 of the homologous chromosomes. Haploid means we have 1. So in meiosis 1, during anaphase, do I pull apart the sister chromatids or do I pull apart the homologous chromosomes? Pull apart the homologous chromosomes. So now I have 1 homologous chromosome in this cell, 1 homologous chromosome in this cell. But I still have a sister chromatid, but there's still just 1 homologous chromosome in each. So now this is my reductive division. Now the cell is the cell is haploid. It was diploid, it's haploid, right? I have 1 copy of each homologous chromosome. But I can't really hang out with 2 sister chromatids. That's too much genetic information, right? So what does it do in the next? Then meiosis 2 is just like mitosis. They line up, separate sister chromatids, and I end up with 4 genetically unique haploid cells. Does that help?
Oh yeah.
Meiosis, it goes through the first part and then the second
Yeah,
part?
meiosis 1 then
Oh,
meiosis
okay.
2.
Yeah.
That's what I'm thinking. It's like,
You're like, how does that
there's 3
happen?
different
No,
things.
just meiosis
Okay.
1 and meiosis 2. And meiosis